Hi, everyone. Thank you so much, Jennifer, for the invitation. It's been a great conference, and I've enjoyed myself so much. So I guess in comparison to everybody else who's gone before me, I'm slightly different position. I'm a postdoc at UCLA right now, but I'm going to be starting my own lab at Ohio State soon. So, and I'm going to be talking to you about how dendritic plasticity mechanisms can contribute towards helping us organize memories together. So what do I mean by memory organization? So yesterday, when we were talking about things, it became very obvious that learning or memories are not made in isolation. Intuitively, we all know this. And we take each single memory, we put it into a large, broad framework, and we pull from this framework as and when we need to make adaptive decisions. And this is what makes us effective in navigating our lives. However, while studying memories, we've largely been studying a single memory and how that evolves or reconsolidates. It's been really hard for practical reasons to study how multiple memories are formed and how they might form links to one another. So one example of how we organize memories is, I'm going to use an example from my own life. So this is my husband and I, we went to a trip that we took to Hawaii a few years ago. It was just one of those trips that was magical. We remembered it, and I'm sure everybody has a vacation like that. We started off on one side of the big island, and saw the sunrise, ended up on the other side on the during sunset, and it was just a very great day. Hawaii is beautiful. So a few years later, we were like, we're going to go back, and we're going to hopefully do a redo. This time when we ended up on Hawaii, there was a historic blizzard warning. We didn't get out of our hotel room, and the only time we got out was to rent ATVs and get muddy. So this is our only picture from that trip. And intuitively, again, we all have experiences like that we put together. We can put them together because they happened on the same day or very close in time. We might put them together because they happened in the same location. All of this happened in Hawaii. Or we might put them together because there are some good trips that we can bunch together and some not so great memories that we can separate out. So in order to understand how these memories are organized together, we are starting with how memories are organized close in time. And the idea and hope is that if we understand how memories are organized close in time, we can take these mechanisms and extrapolate it to how memories might be organized in other dimensions. So I'm going to talk about how memories are organized close in time. And the model system I use is mice. So when Denise Kai was in the lab, she came up with this really beautiful paradigm to understand how two memories, contextual memories, can be linked together because they happen close in time. And I'm going to talk about this paradigm again and again. So I'm going to take a few minutes to describe it. It can get complicated, but I'll also give you a cheat sheet. So briefly, mice will see three different contexts, and these contexts are distinct from one another. We know from various studies that mice can easily tell which one they got trained in, which one they didn't get trained in. And mice see either these contexts very close in time or within the same day. In our case, we usually do it 5 hours apart. There's nothing magical about that 5-hour number, just makes our experiments easier. And the other context is see 7 days apart or an entire week before. And we can ask whether these two contexts that are seen close in time are in any way associated with one another better than the context that they saw 7 days before. And we do this by bringing the mice back in a day or 2 days later and then shocking them in one of those contexts. If you shock mice in a context, they're afraid of that context and they freeze in that context. That's our readout of memory. And we can test this memory by putting them back in this context where we shocked them. And when we do this, mice display fear by freezing at high levels in this context. We can also ask what happens to the fear or freezing in a context that they saw 5 hours before but were never explicitly shocked in. When we do that, mice also freeze in this context. So they're transferring that fear. And I want to say it's a transfer of fear, not a generalized expression of fear, because we can put them back in a context that they've never seen ever, and they won't freeze in that context. So it's not that they're just afraid of everything. I'm not going to show you data, but we can also put them back in this context that they saw 7 days before, and they will not freeze in that context as well. So we have a way where mice will freeze in two contexts that they've seen really close in time. One was made fearful, and one was just associated with the fearful context. And they won't freeze in other contexts. So It's sort of a play between generalization, but at the same time, linking of two episodes. So what do we know about this form of linking? Luckily, there's been some work in the last 5-6 years that allows us to say what might be the cellular mechanisms that might mediate this form of memory linking. So one of the things we know that learning does is when animals learn, this learning is usually found in various sparse network of neurons in a distributed network of brain regions. Within these brain regions, usually there's an ensemble of neurons that undergoes plasticity. And one form of this plasticity is a change in intrinsic excitability. So these neurons become more excitable. I just added that in because I wanted to show some electrophysiology data. But, you know, a lot of people have shown this to be true in various brain regions. And when this neuronal excitability is up, we also know, again, through a large variety of studies, that increasing excitability can specifically bias these neurons to be included into a future memory trace. They're more excitable, more likely to undergo plasticity, synaptic plasticity, and hence be a part of a future memory trace. Essentially what happens when you have these two things combined is that if another memory is formed during the period of time when the excitability is still up, this second memory also is likely to be encoded in many of the overlapping neurons. And this overlap, what this means is that when you think of one memory, it also results in recall of another and forms a link. At least that's our working hypothesis. So neuronal overlap allows you to link memories together, at least close in time. What we don't know is if there are any dendritic plasticity mechanisms that mediate this form of memory linking, or in general, whether the excitability changes that we see are really just in the neurons or also taking place in dendritic compartment. Some of it refers to the idea that Cliff brought up of compartment-specific megaplasticity. So we reason that there is likely a scenario where there are localized changes in dendritic branches, which allow facilitation of future synaptic plasticity on these dendritic branches. And if that does happen, then what you would see is, in addition to these memories being encoded by the same neuronal populations, you would also see synaptic changes or allocation of these memories to similar dendritic branches or overlapping dendritic branches. And that is what I'm going to show you today. So we did all of this by basically, again, giving animals two distinct contextual memories 5 hours apart. And we looked whether these memories were going to the same neuronal population. In addition, we also looked at the dendritic branches and saw if the same dendritic branches were activated and what was the nature of spine changes on these dendritic branches. And then we did it again a few hours later on the same dendritic branches and looked at synaptic plasticity at these branches. And I did all of this in a different part of the brain that we haven't talked about so far, a part called retrosplenial cortex. So for those of you who are not familiar with it, it's a midline dorsal cortical structure. It's beautiful for many reasons, but the reason that I really wanted to use it, among others, is we knew that retrosplenial cortex, just like hippocampus, which we knew showed signatures of memory linking, was really important for spatial as well as contextual learning. We've shown it, other labs have shown it, that it's important for this kind of contextual fear memory. We also knew that what are the neuronal changes that take place in retrosplenial cortex when animals learn a single contextual memory. We also know what are the synaptic or spine changes that took place on retrosplenial cortex and where in which dendrites it took place when animals form a single contextual memory. Made my life easier because I can just go back and track those same dendritic compartments 5 hours later or 7 days later and saw if the changes were taking place in the same area. So help me solve the where problem that we were talking about. And then finally, since it's a midline dorsal cortical structure, it's really very easily accessible to invasive imaging techniques like 2-photon imaging. So I don't have to aspirate a part of the cortex out and then see what happens to the rest of the structure. I can easily longitudinally image these brain regions as and when the animals are learning various tasks. But we did have a problem. What we didn't know is if retrosplenial was in any way involved in memory linking. And so that's the first part of the talk that I'm going to show you. We established what happens in retrosplenial when animals form two memories close in time, and whether this is in any way functionally relevant for the animal's ability to link memories. So the first thing we did was basically use miniscopes and measured GCAM6F-mediated calcium dynamics. And we adapted this technique to work in retrosplenial cortex, and the kind of data we get is essentially, we can tell whether any single neuron is activated or reactivated. We can also tell what are the calcium activity profiles, or how and when in response to behavior these neurons are activated. So it gives us a very rich data set. And my imaging paradigm is something like this, and we're going to use a similar version of this imaging paradigm for the rest of the talk. Essentially, animals see 3 contexts. These contexts are either separated by 5 hours, so really close in time. And we've shown previously that these 2 contexts, when seen at such close time intervals, will transfer peer or will be linked together. Or a context is seen 7 days or an entire week before, and we know this context remains independent. So when we do this, we can assess at a very basic level which neurons are really activated when animals are seeing these contexts, and are they likely to be reactivated. And we find that the overlap in the neuronal population when animals see 2 contexts close in time is much higher than 2 contexts that are seen 7 days apart. This is really robust, and you can, we've also looked at, in addition to just whether the neurons are activated, what happens to their firing rate, their coactivity rates, their ability to decode the context, and done a few other experiments to show that this is not just representational drift, but basically animals' ability to code 2 contexts close in time with the same overlapping neuron. And all of those, I'm sorry, say that again, please. This is A and B and A and C. Oh, right. No, thank you. Yes. Yeah. Yeah, so these are 5 hours apart. This should be NC. I've given this talk and I've never noticed it, so thank you. Yeah, so these are close in time and further apart. And this happens in basically any measure that we've looked at, yes.
And can you just give me some details about how you decide that they're overlapping? You know, these calcium transients, all of them, they're trying to frequency, your sampling is probably low. What makes it you consider overlap?
Yeah, so I can, we've struggled with this question, and I don't think there's like one great way, which is why I was saying we've looked at it in many different ways. So one thing we can do is just, is the neuron activated at any point? Because these are, so when you do 1P imaging, you really don't pick up a neuron, or you can't really segment it out unless it fires pretty robustly. And these are also very, like, much older versions of miniscopes. So we know that we're picking up a neuron, it's really firing robustly during that 10-minute window. In addition, what we can do is basically, I'm sorry?
What is robust?
Well, likely something that would indicate a burst of action potentials, because we're not able to, but in addition to that, another thing we can do is ask if a neuron fires selectively in one of the contexts over the others. And if that lack of, if some neurons are not very selective between 2 contexts, close in time or far apart. And many of the neurons fire at high levels non-selectively between B and C, but not A and C, or A and B. You can do this comparison either which way. In addition, you can look at the population neuronal firing activity and try, train the data from 10% of the neurons and say, can we decode which context they are in? And our ability to decode is much better 7 days or even 2 days apart than when animals see these contexts close in time. Their coactivity rates, how well they fire together, also remain very stable, close in time, but not like as we increase the time window. So we've looked at, if you have another better measure, I'm happy to look at that too. Yeah.
It's super tricky. I'll ask you more later, but it's really interesting data. It's always how you draw that line to say it.
Yeah, no, it's a very fair point. We've also done some probabilistic measures. And the problem also is because we don't have all the neurons, right, which you can in sort of immuno setup, or when you're looking at postmortem tissue, you can tell over the 100% of the neurons how many were reactivated. We don't really have those measures, so makes our math a little more tricky.
Yeah, just follow up on that. Are those old neurons or just the secondary neurons? How do you compute it?
Yeah, so here this was driven by human synapsin promoter. So these are going to be all neurons, but we've also done a similar experiment with smaller data set that was CAMK2 only, and I think the data is pretty similar, really. I'm not going to show you. We've also done it in head fake setup with CAMK2, and we didn't find any changes. All right.
Is it the same animal or not? Is it the same animal? One is 5 hours.
Yes, yeah, it's the same animals that we, so it's a within animal comparison. Okay, so once we had this basically tells us that this is something that happens. So neurons, many of the similar neurons are engaged. What we don't know is if this overlap is in any way functionally significant. Jennifer was talking about some data in hippocampus a few years ago where, you see all these nice changes, but they were not functionally relevant for the trace or the eye blink conditioning task that we were talking about. So we really wanted to know that this is an overlap that we can actually manipulate the linking of memories. So yeah, I guess I already summarized this. So we know there's overlap. Can we now manipulate this overlap? And we did it in a few different ways, but the work that I'm going to, or the data that I'm going to show you today is using the CFOS TTA system. And for those of you who are not familiar, CFOS TTA system is basically a bi-transgenic system. It has TTA under the CFOS promoter. CFOS is an activity-dependent marker, so we can tell which neurons were really active under any behavioral episode. And we can turn this system on and off by giving animals doxycycline chow. So when the animals are on this dox chow, the system is shut off. You don't see any labeling. You take the animals off doxycycline, and you can make a production of effector protein. In our case, it's channel rhodopsin. So we tag cells that are active during a particular window, and then we can reactivate them later and manipulate the memory or this ensemble in that way. So we tested a few different things. I'm going to show you one experiment where we ask, what is the window of memory linking? So they link at 5 hours, they don't link at 7 days. What happens in the time course in between? And what we found in a previous experiment was that at 7 days, mice are kind of all over the place. Some animals link, some don't link. But by 2 days, they basically just don't care that this context was seen before. They don't freeze in that context. So we picked this 2-day window, and we said, okay, typically when mice will see these two contexts 2 days apart, one can be fearful, but the other one doesn't acquire any fearful properties. Can we manipulate overlap, and can we do it only in retrosplenial? And can we then make them either fearful of this previously seen context? Can we manipulate this linking in that way? So that's kind of what I'm doing. Mice will see two contexts 2 days apart, and they'll sit in the home cage. At least our control group sits in the home cage. And this gets a little complicated, so I'll try to go slowly. So they sit in the context, and they see two contexts 2 days apart. A few days later, we shock them in one of the contexts. We know that they're going to freeze in this blue context. And then we test them in either the two previously seen contexts or a completely new context. And we want to know, A, do they learn a single memory? Do they freeze? Do they acquire the linked memory? Or do they also freeze in a previously seen context? And is this a specific transfer of fear because they shouldn't freeze in a context that they've never, ever seen before? So what we find is mice do freeze at pretty high levels in the context they're control mice, in the context they were shocked in, but not in the previously seen context or a completely novel context. So we know memories are not linked at 2 days under control conditions. Now, for our manipulation, what we do is we tag any neuron within the retrosplenial, or given the limitations of the system, RSC neurons with channel rhodopsin during the first contextual learning episode. Then we bring the mice back and we reactivate these neurons again, hopefully, and I've shown this before and others have shown that reactivating these neurons basically reactivates this memory. For example, if this memory was fearful, mice will freeze. So we re-engage these neurons, hopefully redriving any plasticity mechanism that takes place when animals retrieve. And then a day later, they see another neutral context. And then we can test them as we did before. What we find is that this basically drives up the fear in the linked context relative to a context that they've never seen, and also relative to the same context in control animals. So just by taking a small set of neurons in retrosplenial and reactivating them on an intervening day, we can now make mice link memories that they had otherwise never linked. All right, so we know not only is retrosplenial important or shows this linking signature, it's also functionally relevant. We've done chemogenetic experiments and a few different experiments that they all kind of show the same data. So I've shown you so far that retrosplenial is a part of the brain that's important for linking of memories. But what we really wanted to know was whether there was any dendritic plasticity contribution to these memories. And I've sort of hinted at this before, and I know Cliff brought this up. We know that based on a few different studies that there are local dendritic mechanisms that can facilitate synaptic plasticity following an LTP event within the same dendritic branches. And we wanted to test this idea that if first memory basically causes long-term potentiation at certain spines, will there be, for example, let's say, and these are hypotheses right now, an excitability gradient or any other biochemical changes that facilitates LTP formation at a nearby spine, and can we see evidence of this in a linking paradigm? So the first thing we did was we basically monitored spine formation in Taiwan YFP animals in layer 5 apical dendrites. And I told you before that we knew what happens following a single contextual memory formation in retrosplenial. And we knew that these apical dendrites gain spines, and they gain spines in clusters. So we wanted to see if the spine addition and this clustered addition was biased to the same dendritic branches or not. So we monitor them under home cage condition and as they see 3 different contexts. And this is a slightly more complicated version of what I showed you before. Mice see 3 contexts, either close in time or 1 or 2 contexts 7 days apart. And 2 hours after each of this context exposure, we bring the mice in and we see what new spines were formed, whether they're formed close to each other, and so on and so forth. Our control group stays in the home cage throughout this period, and we image them at the same time point. So we have a control for time without context exposure. And we ask a few different questions. This is obviously data rich. We ask if the dendrite that gains more spines following one memory or one context exposure also gains more spines following a second context exposure close in time. We did find that the number of spines that were added to a dendritic branch were highly correlated. And a slightly better way to look at it is by looking at the mutual information in the number of spines added. And so these are basically, these box plots are z-scores of what you would expect if you shuffle the number of spines added to one of the dendrites. So it gives you if there was no relationship between spine addition. And at 5 hours, we see that many of the dendrites that gain spines following one context exposure also tend to gain spines following second context exposure. And the second question, which I think is probably more important, because we know clustered spines are really stable spines and they're important for contextual fear memory, we asked if a dendrite gains a clustered spine, is it more likely to gain clustered spine following a context exposure 5 hours later? So again, we do some shuffling analysis, and these histograms that you see are basically what would happen if this was all happening by chance. And we find that 5 hours apart, when you see when mice see 2 contexts, the percent of clustered spines on a dendrite that's already had clustered spines is much higher than chance. And this is not the case when you're looking at a similar number 7 days apart, even with context exposures. And then finally, we asked, what is the average distance between these newly formed spines? Are they really likely to be in each other's vicinity, close to one another? So when we measure the distance between these spines, which are formed either 7 days or 5 hours apart, we find that, again, the average distance between 2 spines formed following 2 context exposure close in time is much less, around 5 micron, which is sort of a magic number if you look at clustering studies. 5 to 10 microns seems to be the spatial zone where spines or clustered spines are interacting. And otherwise, the distance is much larger. And all of these 3 things, whether the spines are being added to the same dendrite, so the clustered spines are being added to the same dendrite, or whether the distance between these spines is smaller, all of this seems to happen only in conditions where 2 contexts are seen close in time. It does not happen when you either see 2 contexts 7 days apart, or when we image at the same time intervals, either 7 days or 5 hours apart without a context exposure. So it is not just an effective time, the context exposures do drive these spine formation to the same dendritic branches. Okay, so then we also wanted to find out, are these dendritic branches more likely to be activated? And for this, we had to use 2-photon calcium imaging. And for anybody who's interested in dendritic calcium imaging, it has a lot of caveats. So we try to, I'm happy to talk about controls that we did to figure out whether, what we are seeing is really localized or not. But what we did do was we were very agnostic to what is the nature of these calcium signals, because I don't think we have enough information to directly say what they are. We're just saying that these dendritic branches showed us calcium transients. So with that caveat, we were able to isolate single dendritic calcium transients on apical dendritic branches. And because they're mice are head fixed, we had to create or recreate contexts around them. So mice are head fixed, and we create 3 distinct contexts around them. And we're able to track the same dendritic branches across weeks actually. And
what layer?
So it's layer 5 neurons, but apical dendritic branches within layer 5 neurons. So it's the same dendritic branches that we imaged the spines on. Layer 1, yeah. So the dendrites live in layer 1, yeah.
Do you know if they're 5A or 5B dendrites?
I would say that we try to make it both, but it's probably more 5B, because we wanted to be able to make sure we didn't bleed over into layer 4. Yeah. Okay. So we again asked the same question: are many of the same dendritic branches activated? And found out that indeed this is a really robust effect. Many of the same. There's a higher overlap between dendritic branches activated close in time versus further apart. We can also measure their calcium transients and say how many calcium transients were there, and if these calcium transients are more likely to be similar to a dendritic branch that was seen close in time. And indeed, we find that the correlation is much higher. So if this was a proxy of excitability or number of transients in the dendrites, they remain very, very similar, close in time, but not far apart. So these are probability of having a calcium transient. How many calcium transients did they have? And that could be a proxy for activity / excitability.
And your point is dendritic ensembles, but do you know that they're dendrite-specific transients? Or how do you separate them from that?
Yeah, so it's a little bit tricky, so I can show you some of the controls we did, but one thing we can do is basically correlate dendritic branches with each other, and how correlated is the activity. It's very correlated, we can cluster them together into one branch, or say that this is probably likely to be one dendritic branch coming from the same source, whether that's basically because it's back propagating or getting a very similar input, we really obviously can't say. And the size of the cluster tends to be really small. Most average, because we're imaging in a small area and we're doing a sparse injection, and typically the size of that cluster is about 1.1. So there's 2 possibilities. Either we're getting really 1 or 2 dendritic branches per neuron, or we are seeing some heterogeneity in their calcium activity. But I think that those are fairly reasonable caveats to these experiments.
So what you're saying then is just that the same dendrites are active. You're not saying that the dendritic activity is isolated to the dendrites.
No, I don't think I can say that. You're right. Okay, so to the last experiment I want to show you, we showed you that at the neuronal level, we can both observe the same neurons are activated, and we can manipulate the activity of these neurons and hence manipulate the linking of memories. At the dendritic level, I showed you 2 different ways of sort of asking the same questions. Are the same dendritic branches recruited to encode 2 memories close in time? What we really wanted to do was be able to manipulate it, and that's really hard. It's been traditionally hard to not only like isolate dendrites, but manipulate dendritic activity in any reasonable way. And so we had some help from Wanda Ho's lab, and Harao Kasai in 2015 had this really interesting paper where they used the DTE 5' UTR from ARC, which is called DTE, or the dendritic targeting element, and it basically uses this untranslated region to take ARC to recently activated dendrites. We took this untranslated region from ARC and we put it inside our CFOS TTA TED tag system. So now we have, again, a system which we can turn on and off that labels neurons that were recently active in a particular behavioral episode. But in addition to just sending channel rhodopsin all over the cell, this channel rhodopsin in these particular cells only goes to activated dendrites. And again, I'm happy to show you guys controls or some characterization that we did of this system and talk about more that's needed. But we do see very sparse expression of dendritic branches when we do this. We also see no somatic activation or behavioral activation as opposed to channel rhodopsin alone, which goes all over the neurons. And we asked the same question that I've shown you before. Can we take two memories 2 hours apart, and can we reactivate these dendritic branches that we tagged and ask whether two memories 2 days apart that are normally not linked, can we link them now? So as I showed you before, again, animals freeze at very low levels in a context that they've previously seen versus a context that they were shocked in, under controlled conditions when two memories are formed 2 days apart. And this cheat sheet that I forgot to tell you about is we know that there's memory linking when we see this nice V-shaped curve. So we know that the animals freeze at the same level in a previously seen context as well as the previously seen shocked context when we do this manipulation, and much higher than the novel context, indicating that just by manipulating the dendritic branches in one part of their brain, we can actually manipulate memories that were 2 days apart to be linked into the same neurons. I'm almost done. All right, so this is just a summary of what I've shown you, that within retrosplenial cortex, we have overlapping neuronal ensembles that not only encode or are activated following two memories that are formed close in time. We also see that this is functionally relevant. We can manipulate this overlap and hence manipulate linking. And at the dendritic level, again, we see the same dendritic branches undergo synaptic or spine changes. And are reactivated, and we can manipulate finally this overlap at the dendritic level or reactivate certain dendritic branches and hence cause two memories that are formed otherwise 2 days apart and hence independent and link them together. So data I'm not showing you is we're doing some modeling work with Yota Poirazi's lab, and it's been really helpful in figuring out what are the kind of dendritic changes that could underlie this form of interaction between two memories, and what are the plasticity mechanisms that we could potentially experimentally go after. And with that, I want to thank a lot of people. Alcino, obviously, for being an amazing mentor, and I owe him a lot. I especially want to highlight 4 undergrads that have worked with me. They're excellent people. One of them is going to come work for me, so yay. And a lot of collaborators. This has been, wouldn't have been possible without, Wando Ho, Josh, Yota, and a lot of other collaborations that I've had at UCLA. So immense gratitude. And then, as I mentioned, I'm starting my own lab. So if you know anybody who's interested in this kind of work, I'm going to be looking to hire at all levels. Thank you.
All right, questions?
Thanks, such a lovely talk. So you've shown this automatic linking. So what happened? But of course, in the end, they're safe. The animals might learn they're safe in one context and unsafe in another. So do you think that if you then were to train them more, they would learn that distinction? And then what would happen in the breastfeeding?
Yes. Yeah. So I want to rephrase that so I'm sure I understand that. You can cause this linking, and they'll freeze in both contexts. Then you can keep giving them that one context, right? And they will eventually be like, this is no longer fearful. And that can allow them to distinguish these memories. They will still freeze in the linked context, but not the context that you've extinguished them, which perhaps gives us some hints that these memories were always distinct to begin with, but I think there are caveats to that. But yeah, you can at least teach them that these are distinct contexts, and one should be fearful and others shouldn't be fearful.
Do you know what happens in the retrosplenial cortex? What happens in the representation?
No.
Thanks. Really beautiful work.
Thank you.
Over the years, I've developed a love-hate relationship to CFOS, I must admit. So I'm always fascinated by the tagging experiments which have been shown in your system, but also in the hippocampus. I mean, yeah. I think there's truth to it, for sure. So, but I'm still trying to wrap my head around, what do you think is special about the CFOS cells, particularly their activity patterns? Is this, well, it's tagging active neurons, but the learning experience may be rather short in the lifetime. The promoter may be active. And what's your hinge on it? What's your speculation? Why does it work? So what is it picking up?
So let me first not answer that question directly. So we were concerned about some of the similar things, that it might be just something CFOS tag system does randomly, and we do heavily hinge on it. But one other experiment that I kind of alluded to is we do a very similar chemogenetic manipulation. So instead of using the CFOS tag system, we can use the lentivirus, infect a small population of RSE neurons right before both memory, the context A and context B, increase their excitability using CNO or DREADS, and then if these two same populations are excitable, they're both recruited into the engram, and we also see that we can link two memories at 2-day time intervals that way. So it's not necessarily dependent on the CFOS system. We've done a CFOS tag independent manipulation. So that wasn't really the question that you asked. I think that's really fair. I think there's also concerns, like you mentioned, about the drift in the system, right? Because that is why most of our manipulations are done 24 hours after, because the system doesn't stay on much after, as you probably know. But also the neurons that are active on a second exposure and a reactivation are not necessarily the same neurons. There's a lot of difference. And I think those are really interesting questions. The way my take on that and a lot of other things is that clearly there's plasticity during the time of learning that's really important for that learning event. Whether and how that allows long-term memory encoding to take place, I am much less uncertain about. I think the way I like to differentiate it is if you can break that phenomena during the time of learning, it's important for learning, but not necessarily something that's encoding that learning long-term.
Thanks.
Thanks for your talk, and the memory allocation story is really cool. But I'm trying to put 2 and 2 together here. So the idea is that during learning, there's synaptic plasticity, as you just mentioned, but also intrinsic plasticity, which makes the cells more excitable for some period of time afterwards. And that period of excitability seems to be less than 2 days. So when you do the channel rhodopsin stimulation on the intervening day in that previous experiment, so now this is no longer synaptically driven plasticity, or maybe it is, because this is one of my whole questions I have for the whole engram cell story. Anyway, what is actually happening in the network when you're stimulating these CFOS cells with channel rhodopsin? And of course, I'm sure you won't have evidence on some of this, but one of my questions is, if you do continue the calcium imaging after you stimulate with channel rhodopsin, can you see these cells are actually more active, right? At least in some period immediately after the channel rhodopsin, as if there's a sign of intrinsic plasticity kind of being re-engaged as a result of the channel rhodopsin stimulation. I guess that's my main question, which could lead to others, but just as an experimental point, what is the channel rhodopsin stimulation doing, basically?
Yeah, so I think that's probably one of the toughest questions for me to answer, right? Like, I can answer to aspects of it and how that affects the data, but we don't really know what channel rhodopsin manipulations do in any of our systems, right? The whole engram story or anywhere else. I think one way that I would like to, well, at least I like to write about it, if people let me get away with it, which I know is a cop-out, is it's causing a retrieval event, which we know it does because at least animals express fear or various other forms of memory, and that at least it's probably recapitulating some of the retrieval mediated plasticity. It's likely doing a lot more, right? And we don't know. The exact experiment you suggested is something that we've considered. It's hard to do because channel rhodopsin and GCAMP have very overlapping technical reasons, and you can get around them by designing your either GCAMP differently or having a shifted opsin, but we haven't been able to get that to work with a 1P imaging system all put together. So there's like some technical challenges that we can overcome, but I think it's still, every time you artificially stimulate a neuron, whether it's channel rhodopsin or something else, it's probably not doing the exact same thing, and it's a caveat that we sort of live with, and I do too. I don't have a better answer, but...
My other comment is just that I gather from Matthew Larkin's work that the back-propagating action potentials don't make it into the apical tufts anyway. So your calcium imaging there is probably, what's that?
People fight over that. So that's why I was like, I don't know what these potentials are. I acknowledge that. But yeah, some people would say they do backpropagate. Some people say, no, they don't. And I think it's possible, it's probably system differences. The reason we chose apical dendrites also specifically from layer 5 was to get as much distance and perhaps get as much independence from those two compartments. But it's a tough thing to crack.
Yeah, I just wanted to follow up on that because it's such an interesting question whether your spine analysis and your reactivation are happening in the same set of cells. Do you know where FOS expression is in retrosplenial cortex? And let me just say, in many brain areas, layer 5 neurons are very difficult to induce in terms of FOS. So there's lots of FOS expression in superficial layers. I don't think there's much of a layer 4 in retrosplenial. But it might be that you're activating layer 2 neurons, which are doing something to some downstream neuron, say layer 5 neurons, where you see these spine changes. But the FOS neurons and the anatomical changes are happening in a different set of cells.
Yeah, so it's not easy to tell, and this looks really bad over here anyways compared to what I have. But I actually do see, we haven't quantified.
I don't know what that is. Is that
so
fixed tissue histology?
Yeah, fixed tissue histology.
And which way is the midline?
So the midline would be the right side. So this would be the, this would be the midline. So this would be the granular, and this is going to be the...
Right. So very little activation in layer 5.
Yeah, but these neurons are super active. When they are tagged, they tag really, really well. And there is a whole... So either I see very superficial activation, then I don't see anything layer 2, 3, and then I see layer 5 activation, or layer 5, 5A, 5B. So that gives us hope that we're going after the right cells when we're going after the layer 5 apical dendrites. And there are the circuit reasons to believe that these neurons might also be responsible for
I mean, just to push back on that, Rafael Tchaikovsky saw layer 2 neurons that were reactivated. His FOS cells were all in layer 2.
Who's?
Tchaikovsky, he was with Alcina Silva, and he was using the FOS GFP mice. He was imaging, I don't think he was imaging deep layer neurons.
Yeah, so I think that was, those experiments were done in a CFOS GFP line and done using 2-photon. And I think it's harder to also image deeper neurons. And that was also a water maze task, I think.
All I'm saying is I think that based on this histology and based upon what we know about the reactivation of layer 2 neurons, I think that you may have two different subsets of cells that are being differentially engaged by this. The spine changes may not be directly leading to FOS, to Cliff's point.
And maybe Mate, if you want to come get set up, take the last couple questions.
Could I ask you, Mate's sitting up, from a functional standpoint, how do you think about memory allocation at the dendrite versus just onto the same cell? Like, what is the extra functional implication of having that dendritic compartmentalization versus cellular? It seems to me like,
Yeah, and again, this is sort of a little bit of a hand-wavy explanation because null data is really hard to prove. We've tried over the years to link other forms of memories, like context memory with the trace pair memory, different things. And they don't really work very well, but we can do context memory with context memory. Sheena's been able to do a CS memory with a CS memory. And I think one of the ideas that seems that they need to be overlapping dendritic, the inputs need to come onto the similar dendritic branches for these memories to be linked easily. We link all sorts of memories, so maybe we just change where this linking needs to happen, but that might be a functional
might
or
be
a
a
structural
mechanistic
necessity.
thing rather than how it affects the linking in terms of outputs.
Yeah, so we might need this output to be on similar. And this is sort of different than some of the other experiments where people do channel rhodopsin bombardments and reactivate the same neurons.
Great. Let's thank Megha again. Sorry, don't have time.
Thank you.